All right, ladies and gentlemen, today we're going to be talking about RNA and protein synthesis. On this video, I need you to take 4 notes, and I'm talking about the video that's about to play on the screen, not the video that I'm talking in. So take 4 notes over this video, please. All right. So the 4 notes you took, make sure they're on your notes. We are going to check these at the end of this video. Um, our bodies are amazing machines, and we're going to go into how some of this stuff works, how your proteins are made, what RNA actually does. We mentioned it, we talked about it a little bit, but we're going to talk about what it actually does and protein synthesis. So, information in organisms. Okay.
To make a particular protein in the cell, the relevant gene is first switched on in the DNA. A working copy of the gene, called messenger RNA, is made. This copying process is called transcription. Next, the information in the messenger RNA is acted upon to produce a protein. This step.
Okay, so what you get is you have DNA. That's the big dog. That's what controls the production of proteins within a cell. And the steps go DNA to RNA to protein. This is what we call the central dogma of genetics, that DNA starts the process, RNA takes the information and makes it usable to the cell to create proteins. So we have DNA, we have RNA, and we have proteins. Only DNA can lead to RNA, and only RNA can lead to protein. You cannot skip a step in this process. If you do, then you're going to be missing it. Okay? So DNA to RNA to protein. Remember that. That's the only direction, that's the only path we can travel. We can't go from protein to RNA to DNA. That's not possible. We can track proteins as scientists back to where they came from, but we cannot make a protein turn into DNA. So just know the difference there. Now we're going to be talking about RNA. RNA, as you can see in this picture, is very, very similar to DNA. You see the cytosine, you see the guanine, you see the adenine. The only thing that's different in the bases is uracil. Uracil is going to come in and it's going to replace thymine. Okay? So where in DNA we had adenine, thymine matching up, and guanine and cytosine matching up, now we're going to have guanine, cytosine, and adenine, uracil. And now here's another thing that you can see right here on this picture, where DNA is a double helix and we have two strands, RNA is a single helix. Okay? It also stands for ribonucleic acid. Like DNA, it's going to have three main parts, but instead of having deoxyribose as its sugar, it's going to be just ribose. There's no oxygen there, so it's just ribose sugar, a phosphate group, and then a nitrogen base. And again, like I said prior to, RNA contains thymine. Now, most RNAs, especially the RNAs that we're going to be talking about in this class, are single-stranded, okay, and they are in the shape of a helix. Again, just like our rules with DNA, we can take the first letter of uracil and create it to represent what uracil is in our bases. So that's where the U comes in and not the T's. Types of RNA.
Next time, use FedEx.
All right, so what we just saw there was a FedEx commercial. The reason we showed you a FedEx commercial here is because mRNA, or also known as messenger RNA, is going to carry the genetic code or the message that DNA is trying to send from the nucleus to other parts of the cell. If we don't have the message, we can't get the information, right? So this is like the boss telling a manager, okay, this is what I want done, and then the manager coming in and saying, all right, ladies and gentlemen, this is what we need to do, this is how we need to do it, because this is what the boss wants, right? So when this occurs, it takes a message from the DNA and brings it out into the cell. Ribonucleic acid, or rRNA, is actually what's going to read the code. So we're going to take the message from mRNA and it's going to read the code. Next we have tRNA, or transfer RNA. What it's going to do is it's going to transfer the code onto the next amino acid. What it does is it attaches to the messenger RNA with its complementary strand and tells it, okay, that's the amino acid that we need now, and then the amino acid connects in a chain, and that chain grows to form a protein.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
All right, transcription is when DNA is converted to RNA. So transcription, when I tell you to transcribe something, I'm saying write it down, okay? Write what I am saying down. So you're not changing languages. We're not going from one language to another language. We're staying in the same language. We're just changing the way we are communicating. So we're going from communicating verbally to communicating with a written message, right? So RNA is a close relative of DNA. They're the same language. They're still the same language. They're just very different in such a way that it would be like writing as opposed to speaking, or texting as opposed to sending a phone call, right? So the DNA molecule is going to unwind, open up, and be exposed so that the molecule that we just saw went across it can read it. Once this molecule reads it, it uses the base pairing rules we have to go with C. Every time it sees a C on the DNA molecule, it puts a G in that place. Okay? And every time it sees an A, it puts a U instead of a T, right? So the RNA is using A's to pair with T's, and U's to pair with A's, and C's to pair with G's, and G's to pair with C's, instead of A's with T's, C's with G's, right? So we have the same similar things going on, except for instead of T's, we're going to have U's. There's no difference other than a U, okay? Translation now. Let's learn a little bit about translation. This is the process in which a cell uses messenger RNA, what was just created, that long yellow strand, to make a protein. So now we're translating. So for those of us in class that maybe can speak two languages, whether it's Spanish or in English or English and French or whatever it is, this is now taking one language, whether it's written, the written language that we just wrote out, and we're going to transform it into an entirely new language. So it's like going from English to Spanish. Let's watch what this happens.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
All right. So we saw how it worked a little bit. We saw that it created hemoglobin. It creates everything in your body depending on what your body is trying to make. So if you want to make skin color, if you want to make skin, if you want to make nose, if you want to make the shape of your hair, the shape of your eyes, anything like that, the RNA is the ribosome that reads the RNA, which is ribosomal RNA, reads the messenger RNA, and the transfer RNA comes on and creates the amino acid. All of that works together to create all the proteins we have. So genetic coding, here's how that works. You may be asking, how does this happen? How can I figure this out with how DNA is telling my body to make RNA, to make proteins so that I have certain eye color? Well, this is how it works. Amino acids are linked in a sequence in DNA, okay? So DNA makes a code. It's like a secret code that only your body knows for the proteins in a cell by arranging the bases. So it arranges our A's, our T's, our C's, and our G's in a specific order for each protein, okay? For every 3 nucleotides, we get 1 amino acid. Now let me be very clear on this. If I have an A, a C, and a G, that is 1 amino acid that's going to start to build. So to have more than 1 amino acid linked to create a protein, I'm going to need 30 nucleotide bases so I can get 10 amino acids, or 300 so I can get 100 amino acids. It just depends on the protein that we're trying to create. So if I want 1 amino acid, I need 3 nucleotides. So here's an example. The DNA sequence is ATG, CAT, GG, CATA. As you can see, my cursor's coming over the top of this. I did them in different colors so you'd be able to tell the difference here. You got 3 here, which is going to equal, we're going to go ahead and draw on this. This is going to be 1 amino acid. Okay, you can put yourself, if you ever get this question on a test, hint, 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 hint. If you ever get this question on a test, you can put lines for every 3 nucleotides you have. So then I would count. We got 1. We got 2. We got 3. And then we got 4. Okay? So if it has 12 nucleotides, that means there's going to be 4 amino acids. Now there's another way you can do that. You can go 1, you can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and then 12. And then you can divide it because you know that for every 3, we have 1. So you can divide your total number by 3 to get the amount of amino acids that we're going to have. So I got the number 4 right here. And if I do my math the way I just taught you just now, I got the number 4 down here. So that is important for you to know. That is important for you to see that there are 2 ways to figure it out. For Mr. Moore, the easiest way is putting lines in between every 3 amino or nucleotides and then counting the amount of amino acids that I would have in that sequence. Now, when they get up to being like 400, 500 amino acids, that makes it a little bit easier because then you can just see when you cut them down, you can break it up, or you can count them out, count each individual one, and then divide it. So either way, it's kind of a good sequence to use, whether you want to do the math or whether you just want to keep it basic and count 3, 3, 3, 3, and put a line after every 3. Okay. So here's DNA mutations. We talked about this a little bit last unit, but we're going to talk about it a little more here. All right, so bear with me a moment. All right, sorry, my computer froze up, so we're going to go back to that. It's our last slide. But, so there's some mutated animals here. I'm going to go ahead and mute the song. There's some mutated animals that you're going to see here. But what happens is it starts in a base pair sequence change. And this occurs if you have a change in the sequence that you're trying to get done. I'm going to go ahead and pause this after this long-tongued dog. We'll look at the two-faced cat for the rest of the way here. Okay, it starts by changing the base pair sequence. Okay, once these base pair sequences change, the genes, the actual coding for the genes is going to change, and there are causes for this. There's extreme heat, there's radiation, and toxic chemicals. So that's why there's always, people telling you to not stand by the microwave or when you're pregnant, or make sure you're not out in the sun for too long on a really hot day, things like that. When we get mutations, now most of them are not as evident as, let's say, a two-faced cat, but a lot of them are very evident in such a way, like a mutation can cause cancer. Radiation, toxic chemicals, and extreme heat all cause cancer, and that's mostly because of a change in our DNA base pair sequence. Okay, and it's something that we can't really figure out because the change occurs in different areas for all cancers. And then a mutation can sometimes look weird. Sometimes it says most mutations, but most of them don't really look weird. So mutations can, I would go ahead and say exit this out and say mutations can, and put can right here. Sorry, my handwriting's bad on the board, so you know it's going to be bad on here. Can look weird, but a lot of times they don't have an effect on an organism's health. Now, what I just told you about, cancer, of course it can have an effect on organism's health, but not typically. Typically, we're going to have less of an effect on an organism's health, other than the fact that cancer, especially if it's something they're born with. Now, mutations can be extremely damaging. We don't want a ton of mutations. If you have more mutations than not, typically you won't survive. You won't be able to make it. Most of you are 15, 16, 14 years old. So you don't have to worry about that part, but you do have to make sure that you're taking care of your body, protecting it from some of these causes like extreme heat, radiation, and toxic chemicals, so you don't get cancer or cause a defect in your future children, which are down the road with your loving spouses, whether that's a husband or a wife. So that's the end of the notes. If you have any questions, please ask me. Don't be afraid just because this video is out there to come ask me questions. Watch the video again if you don't understand it. Please watch the video again if you don't understand it. Have a wonderful day.